Viva-Voce questions and answers. Compound microscope. Q. What is the main purpose of a compound microscope? A. To magnify small objects or cells invisible to the naked eye. Q. Who invented the compound microscope? A. Zacharias Janssen. Q. What is the magnification range of a compound microscope? A. Usually between 40 times to 1000 times. Q. Name the lenses used in a compound microscope. A. Objective lens and eyepiece lens. Q. What is the function of the condenser? A. It focuses light on the specimen. Q. What is the function of the diaphragm? A. It controls the intensity of light passing through the specimen. Q. What is the difference between coarse and fine adjustment knobs? A. Coarse is for rough focusing, fine is for sharp focusing. Q. What is the function of the mirror in a microscope? A. To reflect light onto the specimen. Q. What type of image does a compound microscope produce? A. Enlarged and inverted image. Q. Why is immersion oil used in microscopy? A. To increase resolution under high magnification. Autoclave. Q. What is an autoclave used for? A. Sterilization of media, instruments, and glassware using steam under pressure. Q. What is the working principle of an autoclave? A. Moist heat sterilization. Q. What is the standard condition for autoclaving? A. 121 degrees Celsius at 15 psi for 15 to 20 minutes. Q. What is the role of the pressure gauge? A. It indicates internal pressure. Q. What material is commonly used to make autoclaves? A. Stainless steel. Q. Why is steam more effective than dry heat? A. It penetrates better and denatures proteins efficiently. Q. What happens if the autoclave lid is not sealed properly? A. Pressure and temperature will not rise sufficiently for sterilization. Q. What safety feature prevents explosion in an autoclave? A. Safety valve. Q. Which biological indicator is used to test autoclave efficiency? A. Bacillus stearothermophilus spores. Q. What type of sterilization does autoclaving represent? A. Moist heat sterilization. Laminar airflow. Q. What is a laminar airflow cabinet used for? A. To provide a sterile, dust-free environment for culturing. Q. What is the working principle? A. Air filtration through HEPA filters. Q. What does HEPA stand for? A. High efficiency particulate air filter. Q. What is the purpose of UV light in laminar flow? A. To sterilize the working area before use. Q. What is the airflow direction in a vertical laminar hood? A. From top to bottom. Q. What is the airflow direction in a horizontal laminar hood? A. From back to front. Q. What is the efficiency of HEPA filters? A. 99.97% for particles greater than or equal to 0.3 μm. Q. Why is laminar airflow used in tissue culture labs? A. To prevent contamination during culture handling. Q. Should the blower be switched on before or after UV light? A. After UV light is turned off. Q. What type of sterilization occurs in laminar flow? A. Air sterilization. Combined pH meter. Q. What is a pH meter used for? A. Measuring hydrogen ion concentration of a solution. Q. What is the working principle of a pH meter? A. It measures voltage difference between reference and glass electrodes. Q. What is the pH of pure water? A. 7, neutral. Q. What type of electrode is used in a combined pH meter? A. Glass electrode. Q. Why is calibration necessary? A. To ensure accurate readings. Q. Which buffer solutions are used for calibration? A. pH 4.0, 7.0, and 9.2 buffers. Q. What happens if electrodes dry up? A. The readings become inaccurate. Q. What is the ideal temperature for calibration? A. Room temperature, 25 degrees C. Q. What is the unit of pH? A. It is a logarithmic, dimensionless number. Q. What does low pH indicate? A. Higher acidity. Hot air oven. Q. What is the function of a hot air oven? A. Dry heat sterilization of glassware. Q. What is the principle of a hot air oven? A. Sterilization by oxidation of cellular components. Q. Standard condition for sterilization? A. 160 degrees Celsius for 2 hours. Q. What is the source of heat? A. Electrical heating coils. Q. Why is aluminum foil used? A. To cover glassware and prevent contamination. Q. Which items cannot be sterilized by a hot air oven? A. Media or heat-sensitive materials. Q. What is the function of a thermostat? A. To control temperature automatically. Q. What is the purpose of air circulation? A. To ensure uniform heating. Q. What biological indicator is used for validation? A. Bacillus subtilis spores. Q. Why is glassware cooled before use? A. To prevent breakage due to thermal shock. Thermocycler, PCR machine. Q. What is the function of a thermocycler? A. To amplify DNA by polymerase chain reaction, PCR. Q. Who invented PCR? A. Carey-Mullis. Q. What are the three main steps of PCR? A. Denaturation, annealing, and extension. Q. What enzyme is used in PCR? A. TAC DNA polymerase. Q. What is the source of TAC polymerase? A. Thermus aquaticus, a thermophilic bacterium. Q. What is the function of primers? A. They define the start and end points of amplification. Q. What temperature is used for denaturation? A. Around 94 to 95 degrees Celsius. Q. What is the function of the heating block? A. To maintain precise temperature cycles. Q. What is the role of Mg superscript 2 ions in PCR? A. They act as cofactors for TAC polymerase. Q. How many DNA copies are produced after 30 cycles? A. Approximately 2 superscript 3, over a billion copies. CO2 incubator. Q. What is a CO2 incubator used for? A. Growing and maintaining cell cultures. Q. What gas concentration is maintained? A. Around 5% CO2. Q. What is the working temperature? A. Around 37 degrees Celsius. Q. What is the function of CO2 in the incubator? A. To maintain pH balance in the culture medium. Q. What is used to control humidity? A. A water tray at the bottom. Q. What type of filter is used for air purification? A. HEPA filter. Q. What is the function of the thermostat? A. To maintain constant temperature. Q. Why is it called a CO2 incubator? A. Because it regulates CO2 gas concentration. Q. What is the function of the display panel? A. To monitor temperature, CO2, and humidity. Q. What type of cultures are maintained in CO2 incubators? A. Mammalian and tissue culture cells. Onion peel observation. Q. What stain is used for onion peel observation? A. Safranine or methylene blue. Q. What type of cell is observed? A. Plant cell. Q. What structure gives shape to the cell? A. Cell wall. Q. What structure controls cell activities? A. Nucleus. Q. What is the function of cytoplasm? A. It supports and suspends organelles. Q. What is the shape of onion cells? A. Rectangular. Q. Why is a cover slip used? A. To protect the specimen and lens. Q. What medium is used for mounting the peel? A. Water or glycerin. Q. What organelle is absent in onion cells? A. Chloroplast. Q. What magnification is usually used? A. 40 times or 100 times. Human cheek cells. Q. What type of cells are human cheek cells? A. Animal epithelial cells. Q. What stain is used? A. Methylene blue. Q. What shape are cheek cells? A. Irregular or polygonal. Q. Do cheek cells have a cell wall? A. No, only a plasma membrane. Q. What is visible under the microscope? A. Nucleus, cytoplasm, and cell membrane. Q. Why are cheek cells transparent? A. Due to lack of pigments. Q. What magnification gives clear view? A. 400 times. Q. How are cells collected? A. By gently scraping inner cheek with a toothpick or swab. Q. Why are cheek cells used for DNA extraction? A. They are easily obtainable and have nuclei. Mitosis. Q. What is mitosis? A. Cell division producing two identical daughter cells. Q. Where is mitosis observed in plants? A. Onion root tip. Q. What are the stages of mitosis? A. Prophase, metaphase, anaphase, telophase. Q. What stain is used? A. Acetocarmine or Fulgan stain. Q. What is the purpose of HCl treatment in the slide? A. To soften root tip tissues for squashing. Bacteria in curd. Q. What type of bacteria is found in curd? A. Lactobacillus. Q. What is the shape of lactobacillus? A. Rod-shaped. Q. What is the function of lactobacillus? A. Converts lactose into lactic acid. Q. What type of microscope is used to observe bacteria in curd? A. Compound microscope. Q. Why is curd considered probiotic? It contains beneficial bacteria that aid digestion.